in my class. In this video, we will talk about the Krebs cycle. The Krebs cycle is also called the TCA cycle or citric acid cycle. This cycle is very important for eukaryotes and prokaryotes to release the energy. The first of all, introduction. The Krebs cycle, also known as the citric acid cycle, TCA cycle, is a series of chemical reactions that take place in the mitochondria of cell. It plays a critical role in the cellular respiration by helping to convert energy storage in nutrients, carbohydrates, fats and proteins into usable energy in the form of ATP, adenosine triphosphate. The Krebs cycle, TCA cycle, is a series of enzyme catalyzed reactions occur in the mitochondrial matrix. They will where acetyl coenzyme A is oxidized to form carbon dioxide and coenzymes are reduced, which generate ATP to a electron form. The cycle name as the Krebs cycle to honor Hans A. Krebs, who discovered it. He discovered it in 1937 and was awarded the Nobel Prize in 1937. It is also known as citric acid cycle. Citric acid cycle is the first intermediate compound of the cycle. This acid has 3 carboxylic groups and hence is also called tricarboxylic acetic cycle, TCA. This aerobic process takes place in the mitochondria with the necessary enzymes are present in matrix. The overall view of the cycle is the acetyl coenzyme A formation, where the cycle begins with the acetyl coenzyme A, which is produced from a carbohydrate like a glucose or fat. Citric acid production to coenzyme acetyl coenzyme A combined with the 4 carbon molecules oxaloacetate with the form of citric acid 6 carbon. The energy is extraction with the citric acid is transformed backward into the oxaloacetate. Through the series of steps, energy is released. This energy is captured in the form of NADH and FADH2 electron carriers. They are the electron carriers. ATP is the energy currency of the cell, and the carbon dioxide is the waste product. They will be released. And the cycle continuation, the cycle continue to oxaloacetate to regenerate and combine with a more acetyl coenzyme A and keep process to go into a new one. The cycle molecules, the Krebs cycle involves several key molecules. These key molecules are the acetyl coenzyme A and oxaloacetic acetate and citrate, and the transformation release energy, carbon dioxide, capturing the high energy electrons, cycles, enzymes, others, catalyzers, and the progression of reaction to convert to the food into a energy. This cycle is complete into only 8 steps. In the different steps. The first one is the citrate synthesis, alkylate, isocitrate, alpha-ketoglucarate, succinyl coenzyme A, succinate dehydrogenation, and furanase and maltane deoxyhydrogenase. This is the Krebs cycle step. The first step, second step, third step, fourth step, 5, 6, 7 and 8. This is the Krebs cycle. That the molecules are make mainly the pyruvate, the metabolism activity of carbohydrate to produce the pyruvate, and acetyl coenzymes A, and they produce the first one is the oxaloacetate. The oxaloacetate again converts with a citrate and isocitrate, ketoglucarate, succinic coenzyme A, succinate, fumarate, valerate, and again they repeat to a oxaloacetate. Now the different steps. The first one step is, in the first step, the cycle acetyl coenzyme A combines with the 4 carbon acceptor molecules and oxaloacetate to form a 6 carbon molecule called a citrate. The rearranged coenzyme A, succinate, fumarate, valerate, and again they repeat to a oxaloacetate. Now the different steps. The first one step is, in the first step, the cycle acetyl coenzyme A combines with the 4 carbon acceptor molecules and oxaloacetate to form a 6 carbon molecule called a citrate. The rearrange in the 6 carbon molecules release 2 of carbon dioxide molecule in the pair of similar reaction, producing a molecule of NADH. And this process, the acetyl coenzymes change to a react with a oxaloacetate acid and convert to a citric acid. Now the step 2. In step 2, there the citrate is converted into the isomer. Isocitrate. This is actually 2-step process involving the first aldol removal and then the addition the water molecules, which is to make a isocitric acid 6 molecule of carbons. Now the third one, in the third step, the isocitrate is the isomer of citrate, which is to oxidize and release a molecule of carbon dioxide, leaving behind the 5 carbon molecules, and they change into a alpha ketoglucarate. Now the step 4th. The 4th step is similar to the 3rd. In this case, alpha ketoglucarate that oxidize and reduce to a NADH and release a molecule of carbon dioxide. In the process, the remaining 4 carbon molecules pick up the coenzyme A, forming a unstable compound, succinyl coenzyme A. These enzymes catalyzing this step, the ketoglucarate dehydrogenase, is also important to regulation of the citric acid cycle. In the fifth step, a succinyl coenzyme A is repaired by a phosphate group, which is then transferred to a ADP to make a ATP or GTP also. If the use of ADP forming of the guanidine triphosphate, and they release the energy. Now the sixth step is the succinate dehydrogenation. In the sixth step, the dehydration process takes place and convert the succinate into a fumarate, and two hydrogen atoms are transferred to FAD, producing the FADH2. The energy content in the electron of these atoms is influencing to reduce the NADH and reduce the FAD. The NADH carries to attach an enzyme and they transfer into a fumaric acid. These steps are found in a mitochondria. Now the seventh step. In the seventh step, the fumarate is converted to a mali. The water is added to fumarate. During this step 7, the meiosis is reduced. That last step in the septic cycle, regenerate oxaloacetate by the oxidizing maltase. Another molecule of NADH is produced. The one molecule of water is added in the fumaric acid in the presence of fumarate to change into a maltase. In this step, they oxidize an oxaloacetic acid in the presence of malic acid, and they produce the NAD and reduce the NADH. 8th number step, that the maltase is again converted into TA oxaloacetate. The maltase is dehydrogenate with the oxaloate, and which combines to another molecule of the acetyl coenzyme A, and to start a again new cycle, the hydrogen removed get transferred to a NAD to form the NADH. This one, the maltase dehydrate catalyze oxalate, and which combines to another molecule of the acetyl coenzyme A, and to start a again new cycle, the hydrogen removed get transferred to a NAD to form the NADH. This one, the maltase dehydrate catalyze reaction. So overall this cycle, the production of citric cycle, there the two molecules of the carbon dioxide release waste, 3 molecules of the NADH, 1 molecule of the FADH2, and 1 molecule of the GTP. These energy carries NADH, FADH2 and ATP to essential for the cell energy production. The conclusion of this cycle, the citric cycle or Krebs cycle is nearly universal central catabolic pathway in which compound derives from a breakdown of the carbohydrates, fats and proteins and oxidize to carbon dioxide. This can be released mostly the energy. The oxidation temporary help to the electron carriers of FADH2 and NADH during the aerobic metabolism. The electrons are transferred and the energy of electrons flow the kept as the ATP. Thank you.